We are now discussing about the chapter molecular basis inheritance. Molecular basis inheritance. Molecular basis inheritance. So, what is molecular basis inheritance? Molecular basis inheritance means the biomolecule. In first piece, we have already studied the biomolecules. Which are the biomolecules? The carbohydrates, proteins, lipids, enzymes, nucleic acid. They are the biomolecules studied in the first PUC. That biomolecules and the molecules are chemicals which are involved in the carried out the biological process in the organism. They are called biomolecules. That a biomolecules involved in the inheritance of characters from generation to the generation. Hence, that is called molecular basis inheritance. That by how the biomolecules will be inherited, that will be a development of the genetic characters in the organisms. Hence, that is called molecular basis inheritance. Now, we will be study out the which molecules will be inherited the genetic characters in the offsprings or generation to the generation. So, the first we see we have studied the one of the biomolecule that is called nucleic acid. Nucleic acid. Nucleic acid is a one of the biomolecule. It will be acts as a genetic material. Genetic material. The transmission of the genetic characters from one generation to the another generation. Hence, that is a genetic material. So that nucleic acid first discovered by the scientist around the Frederick Mischer, and they discovered the nucleic acid. Was they coined the term the nuclein another term one kind ಮಾಡ್ತಾನೆ, and then Altman ಬರ್ತಾನೆ. Altman Frederick Mischer. Frederick Mischer discovered the nucleic acid. It was coined the term nuclein. Then after the Altman they coined the term nucleic acid. That nuclein has the acidic property. That acidic property is present in the nucleic acid that will be discovered by the Altman. Hence, it was called the term the nucleic acid. There are two kinds of nucleic acid. The nucleic acid there two kinds of nucleic acid. There are two types of nucleic acids there. One is the on the base of ribose sugar. A basis ಮೇಲೆ ribose sugar basis ಮೇಲೆ two types of nucleic acids ಅನ್ನು divide ಅನ್ನು ಮಾಡ್ತಾರೆ. One is the RNA ribose nucleic acid ಅಂತ ಹೇಳ್ತೀವಿ. It is made up of a ribose sugar. Ribose sugar is one of the pentose sugar. Then another one is the DNA deoxyribose nucleic acid. It is made up of deoxyribose, a pentose sugar. Both are the pentose sugar. One is the ribose sugar, another one is the deoxyribose sugar. So ribose sugar is a ribose sugar, deoxyribose sugar, both are the pentose sugar C5H10O5. So that is called a ribose sugar. Then a C5H10O4, it is the deoxyribose sugar. This one is the ribose sugar. Ribose sugar. This one is the deoxyribose sugar. Deoxyribose sugar. So deoxy ಅಂದ್ರೆ one oxygen atom ಕಡಿಮೆ ಇರ್ತದೆ ಅಷ್ಟೇ. So that is made up of DNA. A ribose sugar made up of the RNA. RNA is the ribose nucleic acid. It is made up of ribose sugar C5H10O5. The DNA is made up of C5H10O4. That is called deoxyribose sugar. That form the DNA. Both are acts as a genetic material. Only RNA acts as a genetic material in the RNA viruses. RNA viruses ಅಲ್ಲಿ RNA acts as a genetic material. RNA ಏನಾಗ್ತದೆ genetic material ಆಗಿ ಆಕ್ಟ್ ಅನ್ನು ಮಾಡ್ತದೆ. ಆಯ್ತಾ? So RNA acts as a genetic material only in the RNA virus. Which are the RNA virus? Coronavirus, HIV, ಮತ್ತೆ ಟೊಬ್ಯಾಕೊ ಮೊಸಾಯಿಕ್ virus or the RNA virus. In that viruses, RNA can transmit the genetic characters from a generation to the generation. That is a RNA virus. Then all other organisms the RNA cannot be acted as a genetic material. Then DNA acts as a genetic material. In the genetic material means the material or biomolecules which can transfer the genetic characters from one generation to the another generation, that is called genetic material. So DNA acts as a genetic in all kinds of organisms except RNA virus. RNA virus ಅನ್ನಬಿಟ್ಟು including DNA virus ಅಂತ ಹೇಳಿದ್ದು bacteria ಮತ್ತೆ blue green algae all five kingdoms of organisms except the RNA virus ಅನ್ನಬಿಟ್ಟು ಎಲ್ಲವೂ DNA acts as a genetic material. So now we are studying about the how that DNA molecule involved in the inheritance of genetic characters. Then next let us will be the structure of DNA. So what are the chemical composition of the DNA? Chemical composition of the DNA. Chemical composition of DNA. So DNA is one of the type of nucleic acid. It is chemically made up of or chemically contained, deoxyribose sugar. Deoxyribose sugar. It is one of the pentose sugar plus then a nitrogen bases. Then plus phosphate. So these are three chemical compounds are forming the nucleotides. Nucleotides are the a monomeric unit of nucleotides are the monomeric unit of the monomeric unit of a nucleic acid. Nucleic acid the monomeric unit. Nucleotide is the monomeric one unit, the single unit of the nucleotide. So the DNA is a double-stranded polynucleotide chain. DNA is the double-stranded polynucleotide chain. The number of nucleotides are linking one another with a phosphodiester bond. Forming a double-stranded polynucleotide chain. So DNA can be defined as DNA is a double-stranded polynucleotide chain that is made up of number of nucleotides. That nucleotides are chemically made up of the deoxyribose sugar, nitrogen base, and then phosphate. That is the composition of the nucleotide. The nucleotide is the monomeric unit of the nucleic acid or the DNA, and then also RNA. So in the DNA, deoxyribonucleotides are the monomeric unit of the DNA. Next, so nucleotide is made up of a nucleotide is made up of the nucleoside plus a phosphate a nucleoside plus phosphate group together form a nucleotide. So the nucleotide are forming ಒಂದು nucleoside plus ಒಂದು phosphate a phosphate ಸೇರ್ಕೊಂಡು ಏನಾಗಿದೆ ಒಂದು nucleotide ಅನ್ನು ಫಾರ್ಮ್ ಮಾಡಿದೆ. So the nucleotides of the DNA is mainly divided into four types ಅಥವಾ four types of nucleotides ಅನ್ನ ಡಿವೈಡ್ ಅನ್ನು ಮಾಡ್ತಾರೆ on the basis of ಅದರ ಬೇಸಿಸ್ ಮೇಲೆ ನೈಟ್ರೋಜನ್ ಬೇಸಿಸ್ ಮೇಲೆ. So nucleoside ಅಂದ್ರೆ ಏನು? Nucleoside means a nucleoside is made up of nucleoside is made up of a deoxyribose sugar plus a nitrogen base deoxyribose sugar and a nitrogen base form a nucleoside. So the types of nucleotides, four types of nucleotides are present in the DNA. So that is divided on the basis of the type of nitrogen base. So what is the nitrogen bases? That is the nitrogen bases. Nitrogen bases are the organic compounds. They are the organic compounds or benzene ring ಅಥವಾ ring organic compounds. ಸೊ ನೈಟ್ರೋ ಆರ್ಗ್ಯಾನಿಕ್ ಕಾಂಪೌಂಡ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ದೇ ಆರ್ ದ ಮೇಡ್ ಅಪ್ ಆಫ್ ನೈಟ್ರೋ ಆರ್ಗ್ಯಾನಿಕ್ ಕಾಂಪೌಂಡ್ಸ್, ನೈಟ್ರೋಜನ್ ಆಗಿರುವಂಥ ಆರ್ಗ್ಯಾನಿಕ್ ಕಾಂಪೌಂಡ್ಸ್ಗಳು. ದಟ್ ನೈಟ್ರೋಜನ್ ಬೇಸಸ್ ಆರ್ ಮೇನ್ಲಿ ಡಿವೈಡೆಡ್ ಇಂಟು 4 ಟೈಪ್ಸ್. ಆಯ್ತು, ಸರಿ. ಮೇನ್ ಆಗಿ 2 ಟೈಪ್ಸ್ ಇದೆ, ಪ್ಯೂರಿನ್ಸ್ ಅಂಡ್ ಪಿರಿಮಿಡಿನ್ಸ್. ನೈಟ್ರೋಜನ್ ಬೇಸಸ್ ಆರ್ ಮೇನ್ಲಿ ಡಿವೈಡೆಡ್ ಇಂಟು 2 ಟೈಪ್ಸ್. ಒನ್ ಇಸ್ ದ ಪ್ಯೂರಿನ್ಸ್, another one is the pyramids. Right? So the purines are the a larger sized double ring of nitro organic compounds or nitrogen bases. They are the purines are the adenine and guanine. Adenine and guanine. Pyrimidines are the a smaller sized single ring of compounds. So they are the cytosine and thymine in the DNA. So in RNA instead of thymine, the uracil is present. Thymine ಬದಲು ಏನಿರ್ತದೆ RNA ನಲ್ಲಿ uracil, uridine base ಇರ್ತದೆ. So on the base of nitrogen bases ಅಲ್ಲಿ 2 types ಇದೆ. ಒಂದು ಯಾವುದು purines, ಇನ್ನೊಂದು pyrimidines. So purines ಡಬಲ್ ರಿಂಗ್ ಲಾರ್ಜರ್ ಸೈಜ್ಡ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್. ಆ purines ಅಲ್ಲಿ ಎರಡು ಇದೆ, ಅಡಿನೈನ್ ಮತ್ತೆ ಗುವಾನೈನ್. ಇನ್ನೊಂದು ಟೈಪ್ ಯಾವುದು ಪಿರಮಿಡಿನ್ಸ್, ಇದು ಸಿಂಗಲ್ ರಿಂಗು ಸ್ಮಾಲರ್ ಸೈಜ್ಡ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್. ಅದರಲ್ಲಿ ಎಷ್ಟಿದೆ, ಎರಡಿದೆ, ಸೈಟೋಸಿನ್ ಮತ್ತೆ ಥೈಮಿನ್. ಮತ್ತೆ ಅದೇ RNA ಅಂದರೆ ಥೈಮಿನ್ ಬದಲು ಏನಾಗಿರ್ತದೆ, ಯುರಾಸಿಲ್ ಇರ್ತದೆ. ಸೊ ಈ ನೈಟ್ರೋಜನ್ ಬೇಸಸ್ ಅಂದರೆ 4 ಟೈಪ್ಸ್ ಆಫ್ ನೈಟ್ರೋಜನ್ ಬೇಸಸ್ ಇದೆ. ಯಾವ್ದು ಬೇಸಿಸ್ ಮೇಲೆ ಅಡಿನೈನ್, ಥೈಮಿನ್, ಅಡಿನೈನ್, ಗುವಾನೈನ್, ಸೈಟೋಸಿನ್ ಅಂಡ್ ಥೈಮಿನ್. ದೇ ಆರ್ ದ 4 ಟೈಪ್ಸ್. ಆಯ್ತಾ, ಅವನ್ನ ನಾವು 2 ಗ್ರೂಪ್ ಆಗಿ ಮಾಡಿದ್ವಿ, ಪ್ಯೂರಿನ್ಸ್ ಅಂಡ್ ಪಿರಮಿಡಿನ್ಸ್ ಅಂತೇಳಿ. ಆಯ್ತಾ, ಈ ನೈಟ್ರೋಜನ್ ಬೇಸಿಸ್ ಮೇಲೆ ಈ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಅನ್ನ 4 ಟೈಪ್ಸ್ ಆಗಿ ಡಿವೈಡ್ ಮಾಡ್ತಾರೆ. ಆಯ್ತಾ, ದಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಆಫ್ ದಿ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಆರ್ ಇನ್ ದಿ DNA is mainly divided into 4 types on the basis of nitrogen bases. ಆ ನೈಟ್ರೋಜನ್ ಬೇಸಸ್ ಯಾವುದು, ಅಡಿನೈನ್, ಗುವಾನೈನ್, ಸೈಟೋಸಿನ್, ಥೈಮಿನ್. ಅಡಿನೈನ್ ಏನು, ದಿ ಅಡಿನೈನ್ ಅಂಡ್ ಗುವಾನೈನ್ ಆರ್ ದಿ ಪ್ಯೂರಿನ್ಸ್, ಸೈಟೋಸಿನ್ ಅಂಡ್ ಥೈಮಿನ್ ಆರ್ ದಿ ಪಿರಮಿಡಿನ್ಸ್. ಆಯ್ತಾ, ನೆಕ್ಸ್ಟ್ ಎದ್ರ ಬೇಸಿಸ್ ಮೇಲೆ 4 ಟೈಪ್ ಇರೋದು. ಈಗ ಏನು, ಡಿಆಕ್ಸಿ ಅಡಿನಿಲಿಕ್ ಆಸಿಡ್, ಡಿಆಕ್ಸಿ ಅಡಿನಿಲಿಕ್ ಆಸಿಡ್, ಅಡಿನಿಲಿಕ್ ಆಸಿಡ್ ಇದನ್ನ ಏನಂತಾರೆ, ADAMP, ಡಿಆಕ್ಸಿ ಅಡಿನೋಸಿನ್ ಮಾನೋಫಾಸ್ಫೇಟ್, ಡಿಆಕ್ಸಿ ಅಡಿನೋಸಿನ್ ಮಾನೋಫಾಸ್ಫೇಟ್ ಅಂತಾನು ಕರೀತಾರೆ. ನೆಕ್ಸ್ಟ್ ಎರಡನೇದು ಡಿಆಕ್ಸಿ ಗುವಾನಿಲಿಕ್ ಆಸಿಡ್, ಗುವಾನಿಲಿಕ್ ಆಸಿಡ್ ಇದನ್ನ DGNP, ಡಿಆಕ್ಸಿ ಗುವಾನೋಸಿನ್ ಮಾನೋಫಾಸ್ಫೇಟ್. ನೆಕ್ಸ್ಟ್ ಮೂರನೇದು ಡಿಆಕ್ಸಿ ಸೈಟಿಡಿಲಿಕ್ ಆಸಿಡ್, ಸೈಟಿಡಿಲಿಕ್ ಆಸಿಡ್ ಇದನ್ನ DCNP, ಡಿಆಕ್ಸಿ ಸೈಟಿಡಿನ್ ಮಾನೋಫಾಸ್ಫೇಟ್. ನೆಕ್ಸ್ಟ್ ಡಿಆಕ್ಸಿ ಥೈಮಿಡಿಲಿಕ್ ಆಸಿಡ್, ಥೈಮಿಡಿಲಿಕ್ ಆಸಿಡ್ ಇದನ್ನ DTMP, ಏನು, ಡಿಆಕ್ಸಿ ಥೈಮಿಡಿನ್ ಮಾನೋಫಾಸ್ಫೇಟ್. These are the 4 types of nucleotides are present in the DNA based on the nitrogen bases. How they are formed? They are formed by deoxyadenoic acid ಅಥವಾ deoxyadenosin monophosphate made up of a nucleoside plus a phosphate. Nucleoside is made up of the deoxyribosugar and nitrogen base. A nucleoside ಯಾವ್ದ್ರಿಂದ ಫಾರ್ಮ್ ಆಗ್ತದೆ, deoxyribosugar plus nitrogen base ಇಂದ ಫಾರ್ಮ್ ಆಗ್ತದೆ. ಅದರ ಪ್ರಕಾರ ನೋಡಿದ್ರೆ ಯಾವುದು ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಯಾವುದರಿಂದ ಫಾರ್ಮ್ ಇದೆ, ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ಸ್ ಅಲ್ಲಿ ಬಂದ್ರೆ ಗೋನೋಸಿನ್, ಡಿಆಕ್ಸಿ ಅಡಿನೋಸಿನ್, ಪ್ಲಸ್ ಫಾಸ್ಫೇಟ್, ದೆನ್ ಡಿಆಕ್ಸಿ ಅದು ಗುವಾನೋಸಿನ್ plus phosphate. Next deoxycytidine plus phosphate, then a deoxythymidine, thymidine plus phosphate. These are the, a nucleoside plus phosphate form a nucleotide. Nucleosides are formed, deoxyribosugar plus nitrogen base. ಅದರ ಬೇಸಿಸ್ ಮೇಲೆ ದಿ 4 ಟೈಪ್ಸ್ ಆಫ್ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ಸ್ ಗಳು ಇದೆ. ಅದು ಇಲ್ಲೇ ಬರ್ತಾನೆ. ಡಿಆಕ್ಸಿ ಏನೋ ಅಡಿನೋಸಿನ್, ಡಿಆಕ್ಸಿಗೋನೋಸಿನ್, ಡಿಆಕ್ಸಿಸೈಟಿಡಿನ್, deoxythymidine are the 4 types of nucleosides. That nucleosides are combined with a phosphate to form a nucleotide. So, deoxyadenilic acid, deoxyguanilic acid, deoxycytidilic acid, deoxythymidilic acid. These are the nucleotides of the DNA. They are linking one another by a phosphodiester bond to form a double-stranded polynucleotide chain of the DNA. So, thank you.